Hi Z, how are you? Hope all is well with you. That's a nice picture that you have there.
Hey there,
How are you doing?
Katrina.
Have-
Uh, Doing pretty well. Surfing the tsunamis of life and playing with the Chat GPT-3, which did make this piece of art for me in the world.
Oh,
Yeah.
really? Oh, that's cool. That's really
Yeah, this one's not Chat, sorry. Um, this one is the AI art one. So the Chat GP is the for the words, and then this one is from trying to think. I think a program called PhotoFox. So it's generated AI image. So I was incorrect. It's not Chat GPT-3. Anyhow, thank you, Katrina. It's,
well. It's still very cute.
oh,
I
it's
love
so
it.
much fun. It's this, the playing with AI is, I would take me, I just couldn't stop talking about it. I'm having so much fun. Yeah.
Yeah, it is fun. I agree. It's
Mm.
really fun. I tried it when it came out, and then right now I kind of stopped. But yeah, it
Mm.
was a lot of fun trying it out.
Yeah. Well, and so is coming into the Science Society. So this is I just was reading how there's a little write up made, and I was, oh, it's made by science punks embarking on exploring knowledge frontiers with us. And I just love this club and really appreciate the array, like the just the array of people and topics and conversations and friendship that's here. So, like, bravo and thank you, my dear
Oh,
friend.
thank you so much, Z. It's always nice. You're always so kind. But yeah, I think yeah, that makes it interesting, to have so many different people working on so many different things.
Mm-hmm.
Makes
It's like a
me
color wheel.
hopeful. Yeah, exactly.
I, and I hang out in the audience a lot and just listen because that's just my style. So, but I support this room immensely and madly. So I hope that whatever's happening today is goes well, and I'm gonna kind of tuck myself back downstairs and let you do your thing.
Thank you so much, Z. It was really nice talking
Yeah,
with you.
you too.
Thank you.
Meow.
It's so cute. I love it. Yeah, welcome everyone. We will start in around 5 minutes. Um, the presentation is on top, and I also shared the paper and then the lab website. I really recommend checking it out. There's so much interesting work in there that Dr. Elinav did so far and is working on. One I thought was also it was really hard to decide what topic to invite him on. So his talk is broader, um, to explain it in a really interesting way. But there's really interesting microbiome work there. One is he discovered how the mechanism is when people stop smoking, why they gain weight, and even if they have a healthy diet. And he discovered that the microbiome is involved in how the microbiome kind of switches to a different uptake of calories and how that works. I really recommend checking out the lab website if you have time. And yeah, there's a publication page where you can click on all the different works, and the summaries are usually written in a way that people can understand pretty well. So yeah, I'm really looking forward to this discussion because now we read a lot about the microbiome and how it influences gaining weight and all kinds of mental health and all kinds of aspects of our life. But when people like Dr. Elinav started, it was really not something people really believed in too much. I remember when a colleague of mine did her PhD thesis in the UK in a lab that was also kind of pioneering the microbiome work, and the response of psychiatrists was like very that you can basically disregard this. So yeah, it was a lot of great pioneer work that happened in the field that brought us to the knowledge that we are in now. It's thing field, and really looking forward to Dr. Elinav's talk. And he's also new to Clubhouse, so I think we figured most of the technical stuff out. So we'll be fine. Oh, there. Hi, hi, Ran. How are you? To unmute, can you hear me? Okay. So to unmute, there is a little microphone button on the bottom right hand, and if you click on that, we will be able to hear you. If it's the very first time you are joining a room, could be that the unmute function doesn't work right away. So a way to get around this is to just rejoin the room. So to get out and come back in again. Yeah, Iran, I sent you an email asking if you can hear me, and I also wrote in the chat, which doesn't help you much right now. But yeah, if that doesn't work, it's just restarting the app or just joining again. The other thing is, okay. The other thing is that university Wi-Fi's tend to block the interaction. Yeah, because I know that the Weizmann Institute, we had that with guest speakers from the institute before they block social media interaction. So if you could switch to cellular data. Okay, let's see. Okay, we will be figuring it out shortly. Professor Iran will switch to his cell phone and then it should be just fine. Yeah, we had guest speakers from the Weizmann Institute before, and I remember that. Oh, can you hear us now, Iran? So the unmute button is all the way on the bottom right. There's a little microphone symbol, and if you press that,
You should hear me now.
yes, perfect. I remembered that we had months ago from the Weizmann Institute a few guest speakers, and that we had that issue before. Some institutes just you can go on the app or on the social media app, but then you're not allowed to interact. It's a very specific way of blocking, so I just remember.
Okay.
So yeah, and then the slides, since it's not the screen share, it's fine if you have them on your desktop separately.
Yeah, I do. Don't worry.
Great. First of all, thank you so much for coming. It's really an honor having you here. I was just telling the audience about your different type of work you have and how impressive it is. All the different mechanisms you showed, even with quitting smoking and gaining weight, and all the different work you did. It's really very impressive. So we're very honored having you here. And before we start, let me give the audience a short introduction so they get to know you a little bit better. And then we go from there, if that's okay with you.
Yeah, sure.
Okay, so welcome everyone to Science Society, and of course a special welcome to Professor Aaron Elinav. I shared the lab website in the chat for everyone, and also the paper link. Professor Aaron Elinav is at the Weizmann Institute of Science, and he did his Doctor of Medicine at the Hebrew University of Jerusalem, and his his PhD at the Weizmann Institute of Science, and then he did the postdoc at Yale University. When then later he did he had a senior fellow position at the Canadian Institute for Advanced Research, and then he was also the division director of the German Cancer Research Center, and later, what he still holds today, he is the primary investigator at the immunology department, the Weizmann Institute of Science and Research Scholar at the Howard Hughes Medical Institute until today. And Dr. Ran Elinav, he is a researcher that is focused on studying the microbiota, and he is he received the Rappaport Prize for Biomedical Research, the Levinson Award for Basic Science Research, and the Landau Prize of Immunology. And we usually start with a short interview, if that's okay with you, to get to know you a little bit better in a more interactive way. So our first question is, how did you become interested in becoming a scientist and doing research? Was it something you always wanted to do since childhood, or is there something that's kind of sparked your interest if you look back? Thank you.
First of all, thank you for inviting me, and apologies for the technological hiccups. My path was a little bit unusual because I started as a clinician. I studied medicine, and then I I was a resident and a fellow in gastroenterology, so I kind of visited the microbiome every day in my clinical practice. But to be honest, I got a little bored from scoping people, and so I developed a passion to understand better the diseases that I was studying, which led me to a very drastic shift in my career. And I chose to follow both immunology, which was very relevant to many of the inflammatory and neoplastic diseases that we were clinically treating at the time, and also the microbiome, which we were visiting on a daily basis, but regarding as a waste product, which didn't make a lot of sense to me. So it was this curiosity and quite a lucky timing that led me to study the microbiome at an era in which this field was actually just born. So I was there from the start, luckily, and able to kind of follow this field right from its rebirth in 2006.
Yeah, it's really interesting, and I remember when a colleague of mine did her PhD in a lab that started also in this field in the UK, and how when she came to present from our program, how like psychiatrists kind of laughed it off when she first presented. So I was mentioning to the audience how pioneering the work was, because now we hear it all the time in the news, but there was a few years ago, not so long ago, it was very different. So yeah, thank you for bringing us back there, and it's interesting. It's an interesting path, and it's really interesting to learn how the different scientists that come here, how they chose their path. I think it's really interesting. And then you already alluded to how you chose also the specific field you're in. If you could tell us like some peek behind the curtain, how this specific project came about, using phages to basically combat the bad germs, kind of. Thank you so much.
Yes, so as I will shortly allude to, we try to contribute towards a new mechanistic and causal understanding of how our body interacts with the microbiomes in it and around it. And in doing so, already a decade ago, we started to uncover therapeutic targets and therapeutic strategies that could enable to manipulate the microbiome in ways which could be potentially very important for disease prevention and disease treatment. And these include a growing variety of interventions that take or that exploit the fact that, in contrast to our human genome, which of course is very important, our microbiome genome is much greater, but in contrast to the human genome, is amenable to change and to manipulation in the forms of dietary interventions and others. But one of the biggest unmet needs in the field was that most of these therapeutic attempts involved the addition of microbes or microbial products or whole microbiome configurations in the form of fecal microbiome transplantation into an already existing ecosystem, while there was no way to take out or to suppress from this ecosystem microbial strains that were shown to be associated or even contributing to disease. And antibiotics are a horrible way to do this because they're non-specific. They kill off many unrelated bugs. They induce the emergence of resistant strains, and they have a lot of many adverse effects. So this constituted a huge unmet need in the field, and coming up with the first methodology that would enable to very specifically suppress in a silver bullet manner a disease-associated or disease-causative microbial strain in this huge ecosystem without impacting the surrounding became a holy grail for us, and this led to many failed attempts. But one of the ideas was to exploit phages, which are the natural enemies of bacteria, as such form of an innovative targeted therapy.
Yeah, thank you so much. And it's beautiful and elegant work. And for everyone, the presentation is pinned on top. You can access it through that link. And yeah, the stage is yours. Thank you.
Sure, and thanks again for inviting me. And I understand that I need to tell you to advance the slides one after the other. So next slide. Um, and you should be seeing kind of our 2 cents on our potential contribution to the field of microbiome research, and what my two labs at the Weizmann Institute in Israel and at the German National Cancer Center in Germany are trying to contribute our new insights towards a mechanistic understanding of host microbiome interactions. And we and others increasingly can demonstrate that by reaching such deep mechanistic understanding, we would be able to hopefully contribute towards new principles leading to microbiome-mediated disease treatment. Next slide. And I'm going to talk shortly about treatment. And to be honest, in 2023, when we talk about widely used microbiome-associated treatment, we mainly talk about three non-specific avenues of therapy. One called prebiotic therapy, which is a nutritional modality made up of fibers aimed at generating a healthy microbiome, whatever that means. A second is fecal microbiome transplantation, which is a brute force way of replacing a sick person's microbiome with a healthy microbiome. And the third are probiotics, which were there before we knew there was a microbiome. And all of these modalities are in many cases not sufficiently evidence-based, with the exception maybe of FMT in some hospital-acquired infections. But next slide, but the more encouraging aspect of this maturing field is that with the explosion of microbiome research, we are able to contribute towards a number of new avenues of data-driven and more scientific rigorous treatment, at least experimental treatments, and we've contributed to personalized nutrition, to forms of microbial transfer, which are more evidence-based than your over-the-shelf probiotics. These are called precision probiotics, towards metabolite or small molecule treatments that bypass the need to understand the microbes that secrete them, and in manipulating the host side of host microbiome interactions. And I'm not going to talk about all of these today. I'm going to talk about one huge unmet need, as I've mentioned before. Next slide, which is what we try to do when we encounter a microbial species or strain that is associated or even proven to be contributing towards a disease. How do we get rid of it? And to be honest, we have zero means of doing so, and antibiotics are quite poor way of using this suppressive treatment. So this unmet need, close to 7 years ago, triggered our imagination, and we wanted to contribute towards this unmet need. Next slide, and we chose as our model disease, inflammatory bowel disease, which I'm sure is not strange to many of the people in the audience, given the fact that on the one hand, there is overwhelming evidence that the gut microbiome contributes causally to IBD pathogenesis, both in animal models and in human studies. But to be, again, very honest, in 30 years of IBD research, we've quite miserably failed to identify reproducible IBD microbial signatures that are causally connected to the disease. We have no effective human microbiome targeting therapeutic approaches, and still in 2023, IBD treatment still consists mainly of non-personalized and is treatment which is limited to downstream adaptive immune targets. So to next slide, I hope you see my 4 postdocs, Sarah, Denise, Rafa, and Sharon. These amazing 4 postdocs 7 years ago took upon this huge challenge in trying to identify IBD strains in humans that contribute towards inflammatory damage in these patients, and developing the first ever targeted therapy that would enable to suppress or even eradicate them. And next slide. And then first big question that we had to tackle is whether we could identify a human commensal, or as we call it, pathobiome commensal that enables to cause disease in some context, which is associated with worsening IBD across geography. And I emphasize the geographical concept because many of our microbiome studies over the last decade are limited by the fact that the microbiome is highly variable between people and is impacted by factors such as ethnicity and diet and many other environmental factors. So findings that were found to be relevant to one geographical location were not reproducible in other geographical locations, and to find a common denominator strain or strains that contribute causally to IBD necessitated, in our view, to look across more than one geographical location. And next slide. So we've assembled a large cohort of IBD patients and controls in 4 different countries, in France, in Israel, in the US, and in Germany. And we deeply sequenced them for their microbiome using a modality called shotgun metagenomic sequencing that enables to look very deeply into the species and strain level and also into functionality. And when we did so, the first thing that we saw, which was very comforting to us as a sanity check, was that the microbiomes of both main variants of IBD, which is Crohn's disease and ulcerative colitis, were substantially different than those of healthy controls as one big group. You can see on the left the alpha diversity, which represents the diversity or the number of different strains in a given microbiome, and it was lower in IBD patients as compared to controls. And on the right side, you can see the beta diversity, which represents the entire complexity or the structure of the microbiome, which again was substantially different in human IBD patients as compared to controls. So this was true across the entire cohort. And next slide. But importantly, when we looked at each of the 4 cohorts separately, we saw exactly the same thing. So the microbiomes in IBD patients in both variants of IBD were different as compared to controls that were assessed in the same country and assumed to consume similar diets and so on and so forth. And on the right side, you can see that we were also able to look at the functions of the microbiomes by this next generation sequencing method, and to generate a functional signature of the microbiome, which was substantially different in IBD patients as compared to controls, and was consistent between the different geographical locations in diseased or healthy individuals. Next slide. And using this very extensive and deep microbiome probing, we were able to computationally generate a short list of bacterial strains that were very substantially overrepresented in IBD patients as compared to controls across geographies. So you can see here our finalists, our final short list of species that were overrepresented across the different geographies in IBD patients as compared to controls. And you can see that our number 1 candidate of these very interesting species is Klebsiella pneumoniae, which was very substantially overrepresented in IBD patients as compared to controlled across geography, and was also overrepresented in patients that were suffering from an acute exacerbation or flaring of their disease as compared to patients in remission. So all of these merit investigation, but as a first ever development of a suppressive treatment, we chose to focus on Klebsiella pneumoniae. Next slide. And you can see here in these respective volcano plots from each of the countries that indeed Klebsiella pneumoniae was significantly overrepresented and expanded in IBD patients of the different sorts as compared to controlled across different countries, as compared to the local healthy controls in each of the countries. Next slide. Now, you may ask yourself, so if Klebsiella pneumoniae, as an example, is an IBD or a putative IBD causative agent, why don't you just treat the patients with antibiotics? And in addition to all the limitations of antibiotics that I've mentioned before, we wanted to look at our data and specifically to look at the antibiotic resistant repertoires of each of the microbiomes in each of these patients and control across the different countries, And what we found was that IBD patients featured a very marked antimicrobial resistant landscape as compared to healthy controls. In other words, their microbiomes or their microbes were very resistant to antibiotics in general and were carrying many antibiotic resistant genes of many sorts. And this very significant difference in antibiotic resistance was driven by the family to which Klebsiella pneumoniae belongs to. Next slide, and we can even go down to the genes represented by the family, which is called Enterobacteriaceae, and by Klebsiella pneumoniae itself, and we could identify multiple mobile genetic elements and antibiotic-resistant genes that were dramatically overexpressed in these microbes in IBD patients as compared to healthy controls across the different geographies, meaning that in addition to all of the limitations of antibiotics that I've mentioned before, these microbes were highly antibiotic resistant, which would mean that antibiotics are not a viable option in this case. Next slide. But what we really wanted to do is to use the deep genomic data that we've generated in order to reach towards a strain level specificity. We are great believers that going to the resolution of strains below the resolution of species would enable us to potentially identify clusters of microbes of the same species that carry virulence factors that would contribute to their function in different diseases. And when we did so in hundreds of different samples, we were quite surprised to find a clade or subgroup of Klebsiella pneumoniae strains that were overrepresented in IBD patients as compared to controls. Not only these members of this clade were represented, but it was quite obvious. We call this clade Kp2, and this is a highly IBD-associated clade of Klebsiella pneumoniae, and these strains, which are very genomically similar to each other, became our prime suspects as the contributors or the potential contributors to IBD in this context. Next slide, and we've even validated these observations using specific quantitative PCR probes that were developed to identify only these Kp2 strains, and indeed we could see that they were enriched in IBD patients and in IBD patients that were featuring clinical exacerbations as compared to patients in remission. Next slide. So this told us that Klebsiella pneumoniae and specifically the Kp2 strains are strongly associated with IBD. And as I mentioned at the beginning of my talk, we pride ourselves as trying to tackle what I consider to be probably the biggest challenge of our young field, which is to move from correlation and association into causation in trying to show that microbes don't only associate with disease, but actually contribute mechanistically into disease. So our next question is, can we show whether Kp2 strains causally drive intestinal autoinflammation? And next slide, to achieve this task, we performed a very large set of animal experiments in mice using many different strains of what we call germ-free mice, which are mice that are housed in specialized isolators, and these mice are sterile. They don't have any microbiomes of their own, which enables us to transfer into these sterile mice either single microbes or whole microbiome configurations in trying to study whether human microbes impact disease manifestations in this preclinical setting. So for the sake of time, I'm just going to show you a couple of examples of these experiments. The first experiment utilized germ-free mice that were reporting a very central anti-inflammatory cytokine called IL-10. And in these mice, we've to achieve these experiments, we first isolated hundreds of Kp2 or non-Kp2 Klebsiella pneumoniae strains from hundreds of patients in our various cohorts. And then we transferred these individual Kp2 or non-Kp2 strains into these reporter germ-free mice to see whether they would induce pro-inflammatory cytokines or anti-inflammatory cytokines. And the important feature is the ratio between the pro-inflammatory to the anti-inflammatory response. And to make a very long story short, what we could demonstrate is that the monocolonization with a number of Kp2 strains from human patients induced a very small anti-inflammatory response manifesting as low levels of IL-10 and a very potent pro-inflammatory response of the Th1 response, which is characterized by interferon gamma secretion, and the ratio between the two was greatly pro-inflammatory in mice that were monocolonized with these Kp2 strains of IBD-associated microbes as compared to non-Kp2 strains, leading to the conclusion that these strains could contribute to the immune response that contributes or even drives IBD in patients. Next slide. A second example involves germ-free mice that lack this anti-inflammatory cytokine IL-10, and these mice under colonized condition are considered one of the leading models for human IBD in mice. So in the absence of this anti-inflammatory cytokine, these mice spontaneously develop signs of autologous inflammation in the gut, which lead to tissue damage and manifestations that mimic human IBD. Now, these germ-free, these sterile IL-10 reporter mice, knockout mice, not reporter mice in this case, were now used as recipients of Kp2 or non-Kp2 strains from human patients to see whether these strains could induce an inflammatory response mediated by these microbiome elements that would mimic IBD, and this was indeed the case. You can see here an example in the middle panel of 3 different Kp2 strains that induced a very potent gut inflammatory response, manifesting as an elevation in a protein called lipocalin in the middle, and in secretion of pro-inflammatory cytokines by T cells in the gut, in the right panel, which is exactly what we would expect in this model of IBD when disease starts. And next slide. And when we looked at the histopathological score, which basically quantitates the tissue damage that results from this uncontrolled inflammation, we saw that the monocolonization with these strains of human Kp2 was able to induce tissue damage that was very similar to what we would expect in a human IBD setting. And we could even almost perfectly correlate the amount of induction of a pro-inflammatory response by these Kp2 strains to the amount of tissue damage that this response was inducing. This is in the right panel. Next slide. So this told us that at least in the mouse experimentation setting, we could demonstrate that Kp2 strains were able to induce an autoinflammatory response in the gut that would lead to tissue damage and features that are reminiscent of human IBD, which brought us to the billion-dollar question. Now that we've proven causality and pinpointed the clade of strains that, at least in this example, contribute towards this causality, can we find a way to specifically and efficiently suppress them and to induce an amelioration of IBD? Next slide. And of the many different things that we attempted and thought of, we chose to focus on bacteriophages. And bacteriophages are very interesting. These are viruses, but these are special viruses that only infect bacteria in a very specific manner. They have receptors and pathways in different bacteria that recognize different phages. And in fact, bacteria and phages are old enemies throughout evolution of each other. So the idea was to use these enemies of bacteria, these lytic phages, as means of suppressing a bacteria of interest. Now, if you move on to the next, if you press enter to the next part of the slide, the big advantages of using phages as an anti-commensal microbiome treatment is that they're very species specific. They have a narrow range of specificity, which would mean that you could potentially target a group of strains without impacting the entire ecosystem that surrounds them. They've been used as form of I would say, a cultural treatment, a popular cultural treatment, mainly in the former Soviet Union for almost a century, for reasons that mainly stem for the high price of antibiotics. So there's a lot of kind of common data showing that they're non-toxic and generally safe for usage in humans, because we are not aware of any receptors for phages that are expressed on eukaryotic cells. and therefore they're recognized as self. Next. But there are also very big challenges of the usage of phages as antibacterial therapies. The first is that if you give phages systemically, phages are recognized as foreign and induced a very potent immunogenic response. So you can only use them systemically for a limited amount of time. And the solution we came up with is to use phages for the first time as oral treatment in order to only impact the microbiome in the gut without having to deal with the major systemic immunogenicity that develops upon systemic transfer of these phages. And next, but the biggest challenge of phage therapy is the concept of phage resistance. As I've mentioned, phages and bacteria are old enemies of each other. Phages are very common in almost all habitats in which you can find bacteria. And in response to these phages, bacteria have developed a very wide repertoire of anti-phage mechanisms of defense, which you know of many of them. For example, restriction endonucleases that are so widely used in molecular biology are in fact bacterial proteins that degrade phages. CRISPR, which we all, or many of us, massively use, is in fact a bacterial system that is able to identify, destroy, and remember phage attacks in order to prevent them in the future. And there are many more new such systems that are being explored. So if you were to give a single phage that attacks a bacteria of interest, there is a 100% chance that the bacteria would develop resistance against this phage. And the solution we thought of coming up with for this major limitation is to develop a phage cocktail in which we would identify different phages that would attack the bacteria of interest through different receptors and mechanisms, and together the bacteria would not be able to develop anti-phage resistance. Next. So this resulted in a very painful and very long process of hunting for phages, mainly in sewer samples. We connected to the national sewer system in Israel, but also from microbiome samples, from dental waste, under the sink, because phages are also very, very resistant to detergents and so on and so forth. And with this approach, we've assembled a very large library of many, many thousands of phages against Klebsiella pneumoniae. And in a screening process, we've subjected these phages to different Klebsiella pneumoniae strains to learn about their specificities and resistant profiles. Next, and this generated these very long and boring Excel sheets of resistance and sensitivities of many different phages. Next, which led to the identification of 48 phages against Klebsiella pneumoniae, which seemed to be the best in the screening process, which entered the in vitro validation. And in this in vitro validation step, we took these 48 phages and we assembled them into cocktails consisting of 3, 4, or 5 phages, and we subjected them into Klebsiella pneumoniae strains that were very highly resistant in different repertoires against different phages. Next, and this resulted in many different growth inhibition results. I'm not going into the boring details, but in this in vitro phase, we identified 5 of the 18 phage combinations which seem to have the best and the widest Klebsiella pneumoniae suppressive capacity when subjected into all of these resistant strains in the in vitro setting. Next. So we at this stage, we had 5 phage cocktails that entered the in vivo assessment process. And in this in vivo assessment process, we took mice, we colonized them with resistant Klebsiella pneumoniae strains of IBD relevance. And then we gave these mice orally the different cocktails of phages to see whether they could feature the same suppressive capacity in the in vivo setting, which is much more complicated and hostile, as we see in the in vitro setting. And next. And this, again, resulted in many phage growth curves, this time when taken from mice. And one of the 5 phage cocktails seemed to be better in its activity against different Klebsiella pneumoniae strains as compared to the other. This was called the 5E. This was the cocktail 5E next. And after this very long process that took us a year to complete, we declared cocktail 5E as our winner. And I'm sure all of you want to meet the winner next. So this is an electron microscopy set of pictures showing you the 5 members of this 5E phage cocktail. Of course, we sequence them very deeply. We understand how they come from different families. We know their virulence factors, and we now know many things about these 5 phages that form the cocktail next. And as a sanity check, we were able to go back into our IBD and control microbiome bacterial databases and to identify that the receptors that recognize these 5 phages were markedly enriched in IBD microbiomes as compared to controls, which was an important sanity check for us. Next, so as a summary of this stage, we uncovered in a very painful iterative process a phage combination that could potentially suppress the Kp2 strains that were contributing towards IBD. But now the burden of proof was on us to try and demonstrate that this phage cocktail would actually suppress intestinal autoinflammation in the preclinical mouse setting. And next. So we started with wild-type germ-free mice. These are, again, these sterile mice. We mono-inoculated them with representatives of the Kp2 strains coming from humans. We gave them the phage cocktail, and as you can see on the right panel, when we only gave them the human pro-inflammatory Kp2 strains in the second bar or the second group of dots, you can see that these strains induced a potent pro-inflammatory response, manifesting as an interferon gamma release from CD4 T cells in the gut, as I've shown you several times in previous slides. But importantly, when we added on top of it the 5-phage cocktail, it not only suppressed the microbes that induced the response, it actually suppressed the response itself. Next. But the biggest challenge was to show that this has an effect on a model of colonic inflammation that mimics some features of human IBD. In this model, we chose a model called dextro and sodium sulfate, in which we give a toxin which leads to uncontrolled inflammation in the large intestine of mice, which mimics several aspects of human IBD. And when we gave mice that carried the Kp2 strains, which contributed towards their inflammation, the 5-phage cocktail, we could see that the phages in this inflammatory setting were able to suppress the Kp2 strains. You can see on the left how they do it in the lumen, and on the right how they can even do it in Kp2 strains that are attached to the lining epithelial cells in the host. These are the bacteria which are most important because they affect the host from a closer range. Next. And the result of this bacterial suppression was a reduction in multiple cytokines that represent the pro-inflammatory response that Kp2 strains induce upon colonization of mice in this DSS model. You can see here some examples. And next, and you can see that the resultant tissue damage was also dampened or ameliorated by the administration of the 5-phage cocktails, as featured by colonoscopy, which we performed in mice on the left, and by histopathological score of sections of the large intestine in the middle and on the right, which featured a reduction in inflammation and tissue damage upon the suppression of the Kp2 bacteria in these mice. Next, and we could even follow the same model for much longer periods of time and assess the potential development of Kp2 strains that would become resistant to the phage cocktail, and we couldn't see such resistance developing, probably because we gave 5 different phages that were acting in harmony to suppress the bacteria. So you can see that the bacteria were significantly suppressed on the left, and in these longer periods of inflammation, we could even see that the 5-phage cocktail was able to reduce substantially the level of mortality of these inflammation-induced mice on the right. Next. So this told us that the phage cocktail, at least in the preclinical setting, was able to suppress the bacteria and, more importantly, suppress the inflammatory responses and the tissue damage that these bacteria were generating. But as a final part of this study, we wanted to go back to the bedside, quote unquote, to see whether we can scrape the surface in demonstrating that such a multi-phage treatment could be potentially used as human therapeutics. And the first thing that we did next is to test our phages in an ex vivo human-like gut system. This is a system which consists of different containers which sequentially contain different goodies that represent the different compartments along the human gastrointestinal tract. So, for example, the containers that represent different features of the stomach are acidic and they have pepsin and so on and so forth. And then the proximal small intestine containers have bile acids and pancreatic juice. And then the colonic ones have a large assortment of human microbiomes and so on and so forth. And this system allows you to pass, in this case, the phages from one compartment to the other and to see what the shifting biophysical properties of the human gastrointestinal tract are doing to the phages. And next, when we did this, we learned a very important lesson, which is that when we pass the phages through the compartments that represent the gastric compartment, which were very acidic, we saw that the acidity substantially reduced the numbers and the viability of the phages that we've administered almost to undetectable levels. So the acidity in the stomach was destroying our phages. However, next, when we counteracted and basically added acid suppressants to the stomach compartment, and the stomach was no longer super acidic, then the phages persisted through this compartment, and following the stomach, the phages persisted in very nice quantities throughout the chambers that represent the different regions of the small and the large intestine. Next, and this led us to the first in human phase 1 clinical trial, in which we assembled a group of healthy young volunteers and tested the two components of the phage cocktail on them. And given the result that I've shown you in the previous slide, all of these individuals were given proton pump inhibitors, which prevent the acidity in the stomach and would enable these phages to survive the stomach and to reach the other more clinically relevant compartments along the human gastrointestinal tract. This was a very special trial because, in contrast to all trials that we've engaged in and probably you've engaged in, we didn't just sample these participants for their stool. We actually collected everything that they've generated, all the stool that they've generated throughout a week of very close follow-up. And you can imagine our -80 freezers, but this enabled us to quantify very accurately the number, the proliferation, the expansion, or the destruction of these phages in a very quantitative manner as compared to what we've given these individuals. Next. The results were that these two representative phages persisted super nicely throughout the gastrointestinal tract of these participants. And in fact, they seem to have reached a concentration in the lower gastrointestinal tract that was 1,000-fold higher than what would be expected to efficiently kill the Kp2 strains if these were IBD patients. So it seems to be at least feasible, and even to some extent self-propelling, because even after a few days that we've given these phages to these individuals and we've stopped, we could see that the phages could persist along their gut, meaning that maybe they were meeting some of their targets and were persistent to some extent for these few days. Next. Of course, we couldn't assess in this first in human phase 1 clinical trial whether the phages would be efficiently eradicating Klebsiella pneumoniae Kp2 strains, because these were healthy individuals. They don't carry any Klebsiella pneumoniae. You can see here the shotgun metagenomic sequencing from these individuals. They basically don't have Kp2 strains as would be expected. Next, but what we could see is whether these specific phages have an off-target effect against unrelated bacterial strains in the microbiome, and we saw that this was not the case. In fact, the microbiome remained stable, and we saw no statistically significant differences in this non-Kp2 harboring microbiome following the administration of phages as compared to the control group. And I forgot to mention, we had a control group that only received vehicle throughout the trial. Next. The story that I try to share with you has very many implications for IBD, but I think it has potentially greater implications even beyond, which pertain to the entire microbiome field with respect to the potential of using a phage-based therapeutic pipeline to develop targeted treatments for many other strains in IBD and for strains that are associated with any microbiome-associated disease that you can imagine. Next, and the pipeline we propose is that one would identify and test human disease-associated strains, preferentially across different geographies and ethnicities, isolate representatives, and test them for causality in the preclinical mouse setting. Next, and then one could generate through an iterative process a phage combination that would target the strain clade of interest. And now this took us years to develop, but now that we know all the stupid mistakes that we've made, which I'm not sharing with you, we think that this process could be expedited much more efficiently than we've done. And with follow-up projects, we are generating such cocktails much faster. Next, and then once you have a phage combination, you can treat it or you can test it in the preclinical mouse setting for its suppressive activity and its disease preventive activity next, and then take it to the human bedside to test it in the form of clinical trials. And next, and in the larger kind of bird's eye view, what we hope would be another kind of tool that we would add to the precision medicine shelf, in addition to the many others which we and others are developing, which I'm not sharing with you today, that would enable to increasingly harness science and data-driven approaches in order to target microbiome elements and treat humans in therapies that are tailored to the individual. This would also enable us to, of course, to use such treatment as a research tool to test the importance or lack thereof of different strains in the preclinical setting. Next, and this brings me to the end of my talk, and I would like to deeply thank the many people from across the world who have joined me for this quite long journey in many different and exciting projects. I couldn't do this without them, and I've mentioned the champions, but many other people are involved. And I'd like to take the opportunity to thank the many clinician teams from across the world and leading basic science teams that have teamed up with us in generating this advance and this project, and the spin-off company that we've generated based on the results of these experiments called Biomix, which is upscaling the efforts in order to come up with new combination phage therapies for a number of human microbiome-associated disease. Of course, all the funding agencies that have made all of this possible. Next, and as of the last 4 years, my second affiliation in parallel to the Weizmann Institute at the German National Cancer Center, the DKFZ in Heidelberg, in which we have a great group of young scientists that are utilizing all the exciting tools that we've developed at the Weizmann in order to study the possible contributions of the microbiome towards cancer development, progression, and treatment responsiveness, which is a completely different story for another time, but I thank them as well, and I think I'll stop here. Thanks.
Thank you so much for this wonderful presentation and for doing all this work. I know you said, it was, very tedious and like a power, like powering through that. But this will, these lists of different phages and all this different data will in the future be so valuable. So this is really wonderful. And I wanted to tell the audience, please share your questions or raise your hand if you have questions. And in the meantime, I'll start asking a few questions. And then if you raise a hand, I'll bring you up. And it's really interesting to see that, the cocktail you use, that it can basically survive long enough to become effective in the future. Like, what's the time span of that you still find phages in the body? Is there, does it at some point ebb down? And if how long does it take?
Yeah, it's a wonderful question, mainly because I don't know the answer. I would say this. First of all, we're talking about strictly lytic phages. We try to avoid lysogenic phages that would affect, for example, antibiotic resistance spectrums in bacteria. We don't want to cause any damage. The advantage, one of the advantages of this combination phage treatment approach is that, at least theoretically, as long as these phages meet their target in the body, they would invade their bacterial targets, propagate within the bacterial cells, explode the bacteria, and from one phage you'll have 1,000 phages and they would be self-persistent. Now, of course, this is in theory true, but in reality, what we think is that this may constitute a live treatment, but it would have to be replenished, and the frequency of replenishment would probably depend on the phage combinations and on the bacterial growth rates and inhibition rates. So it would probably be different from bacteria to bacteria. We've seen some hints in our very first crude human trial for such persistence, but we are not even close to understanding the full kinetics of this approach.
Yeah, that's really interesting because I was imagining you could have this one day as a preventive type of medicine, especially in countries where you have, for example, very high rates of Helicobacter and then related cancer risks. Would that be something that that you were thinking of?
Yeah, absolutely. So you can think of any commensal that is associated with disease, and you can potentially imagine that this could be used as a very safe and easy suppressive treatment that would avoid the need for repetitive antibiotics. So Helicobacter is just one example. Helicobacter is a commensal in the human gastrointestinal tract. It got a very bad reputation being the first, and given its strong contribution to cancer, but it's not the only one by any means. And so you can regard it as a commensal, just like any other commensal, which in some cases needs to be suppressed. And this could generate one platform for such a suppression.
Yeah, that's really interesting and really wonderful. Would phages also be able to attack other things, like not just bacteria? You, you could think of, I don't know, cancer, gut, in the gut or so. Would that one day maybe be possible? Is that something you maybe are thinking of?
Well, the issue with phages, or one of the safety advantages of phages, that to the best of our knowledge, there are no eukaryotic receptors for phages expressed on eukaryotic cells. So this is why they are considered so safe. So thinking of the possibility of direct phage effects on human cells is probably the possibility is quite low. Although there could be, for example, and there have been some studies suggesting that the genomic material in phages, the DNA in phages, could generate sensing by innate immune receptors that recognize foreign DNA. which could lead to some host reactivity, which is indirect and non-specific. What the clinical implications of this are, we don't know, and we estimate that they're quite low. But since we know, for example, that regions in our body that were previously considered to be sterile, such as even tumors, there is a low biomass microbial population in tumors that was discovered just a few years ago, which potentially could lead to contributions to cancer or to impact the tumor microenvironment. One could think of systemic administration of phages that would swim to the tumor and would attack these intratumoral bacteria and therefore prevent or treat features of cancer. And all of these aspects are being explored as we speak.
Yeah, I can imagine. And of course, I would assume, let's say, in elderly patients, there's a really high rate of infections on legs, of wounds, like related to diabetes and so on. So, would that also be something, your company or your lab is maybe trying to address in the future with phages?
Yeah, absolutely. So the concept of phages treating a pathogenic bacterial infection has been attempted and succeeded in some cases before us. Systemic effects of single phage treatments on sepsis and on antibiotic resistant infections that are life threatening are out there as case reports. You can find them and are being developed. So these are time limited attempts given the immunogenicity, but in some cases, very impressive successes have been reported. I think that the novelty of what we do is that we do not target these systemically very aggressive pathogens, but we actually are trying to specifically fish out commensals from a very dense and heterogeneous commensal population in the microbiome without harming the surrounding. So these are two different approaches that could be complementary. You could even think of combining an oral and a systemic phage therapy to not only target the commensals, but also target the commensals which are locally invasive and in some cases could lead to clinical manifestations and improve your treatment. Again, these are things that we and others are checking and exploring as potential future developments of this treatment approach.
Yeah, I'm imagining like instead of an antibiotic ointment, just have a phages ointment for people. But that's probably a little bit, when somebody gets a scratch, like elderly, just have a cream with phages at home. I don't know. Probably it's not possible. Do you need to keep them refrigerated or you
No,
need to keep them?
phages are exceedingly stable. You can find them in soil, you can find them in sewer. That's why we looked for them in sewer and in dental waste, because the bacteria get killed by detergents and so on and so forth, but the phages persist. So wherever you can expect large quantities of human excretions, even when we get rid of the bacteria in sewer by different detergents and so on and so forth, the phages persist, and you would find high concentrations of these phages, and these phages would come from bacteria that are relevant to the human gut. Same for dental extrusions that would be enriched for phages that come or that would attack the oral microbiome commensals. So phages are exceedingly stable. They do not necessarily need to be refrigerated, and therefore they're very convenient for usage.
I'm imagining toothpaste, detergent, everything with phages. I'm sorry. Please go. Dr. Shah, did you have a question?
Yeah, thank you so much, Aaron, for your wonderful talk. So let me starting my question. So how much early you think that we can use the phage therapy, for example, in a pediatric patients? So in the case of the side effects as well as the products that we have, you think that by considering the allergy reaction and all of those side effects that we might see in a pediatric patient, you think how much early we are able to use
Yeah,
that in a clinical?
yeah, it's a great question because on the one hand, vulnerable populations such as pregnant females and children and so on and so forth are the ones that we kind of are most careful in terms of clinical attempts and clinical trials because they are vulnerable. And so I don't know. But on the other hand, the pediatric population is also very attractive from this aspect because the healthy normal configuration of microbiomes that we carry for our entire lives is formed in the first 2 or 3 years of life. This is the window of opportunity, and impacting a disease-causing or disease-associated microbiome configuration at this window of opportunity would be expected to be most efficient. So the answer to your question is we don't know, but we think we definitely need to carefully but persistently study this because of all the different microbiome interventions, I would guess that phages could be of the safest interventions, but this is just speculation land, and we need to do the experiment.
Also, so by considering that you just mentioned about the pro-inflammatory as well as all of those interleukin that they are involved, include the regulatory, which you just mentioned, I think, as part of your slide, that was IL-10, if I'm right. So if you want to consider in some of the condition, for example, like glioblastoma, again, I'm asking about the pediatric. And as you know that when we are thinking about the cancer environment is totally different area, but however, phages, they are very favorable for, like a cargo and moving the drug to the targeted point actually. And for example, glioblastoma, you think that how much they can be beneficial in that early age? Do you have any kind of hypotheses based upon your research?
Yeah, it's a fantastic question. I have absolutely no clue. One could think of two options for a cargo feature. One is of the bacteria themselves, right? Because I've mentioned that we increasingly know that tumors harbor a very low biomass microbiome inside them, which is specific to different tumors. And if we understand how the microbes got there, or if we could even give the microbes, the bacteria systemically and expect that they would preferentially concentrate in the right tumor, then one could use the bacteria as a kind of a cargo, a cargo-loaded treatment that would reach the tumor preferentially. That's one speculative but researched possibility. The second is to use the phages against bacteria. So to target the phages against bacteria that we know are concentrated in the tumors, and then the phages would home into the tumor through their recognition of the bacteria and would release their cargo. These are very fantastic possibilities. Again, we need to do the experiment, but these are of many different options that these discoveries develop or make possible in future projects.
And you prefer the conjugated therapy in comparison with the monotherapy, right?
Yeah, I mean, it really depends on what your purpose is. If you don't care about phage resistant developing, you just need to carry some radio labeled or chemotherapeutic or immunotherapeutic agent to the tumor for a short period of time, then you could give a single phage. But if you really want to generate a more long-lasting effect, especially in suppressing bacteria, then I think at least currently, there's no other way than using the combination phage therapy that we've developed.
What about the duration by considering the mono or conjugated?
Again, if you take it to simple terms and you consider an in vitro bacterial colony consisting of 1 million copies of whatever, E. coli, and you would give a single phage, there's a 100% chance that one of the million copies of E. coli would spontaneously have a mutated receptor for that phage. So there's a 100% chance that resistance would develop to one phage. Now, the more phages you add on to this equation, the lesser the possibility of a bacteria developing resistance. The duration, I don't know. I have no clue, to be honest. You know, all I can say is that it probably would change between different combinations and different bacteria, depending on the pharmacokinetics of both components. But this is something that we would definitely need to explore in future trials.
And what about the route? For example, PO, IV, nebulizing, what type you think? What kind of route?
You know, depending on what you want to target, if you want to target commensals in the gut while avoiding the immunogenicity, then the oral route would be the most favorable. This is what we've tried in this project. If you also want to target locally invasive commensals, such as we know exist in primary sclerosing cholangitis and other associated diseases, then you would probably need to combine an oral and an intravenous therapy. If you want to target a pathogen that is systemically dispersed in sepsis or in systemic infection of many sorts, I don't see any way to do this without giving the phage systemically. So it really depends on where you find the bacteria, and that really determines which route you would use.
Thank you so much for your answers, Eran. I'm passing the mic to Katerina.
Yes, thank you so much. And I want to be respectful of your time. We have been going a little bit over an hour. So thank you so much for coming here, presenting, and answering our questions. This was really a really interesting discussion, and it's such an interesting and important work. So we are curious to follow you along in the future, and maybe we hear you back one day to talk about your cancer research. Thank you.
Bye, thank you.
Yes, thank you so much. And thank you everyone for coming, asking questions, posting comments in the chat. Yeah, we are really honored that Professor Elinav, that you were here today. And if you like discussions like this, just follow the club. The next one will be with Dr. Chu from the Brookhaven National Lab, from the Particle Collider, the Star Alliance. He will talk about the new type of quantum entanglement that was used to see inside of nuclei. So it's a different type of discussion, but I think it will be also interesting. So thank you so much, Dr. Elinav, and we wish you all the best for the future. It's totally egoistic to wish you all the best, and I'm looking forward to have the toothpaste with the pages. No, I'm kidding. Okay, thank you so much, everyone. I'll close the room in 3, 2, 1. Bye, everyone. Thank you.